Imagine the dodo, extinct for 3 centuries due to human activity, walking again through genetic engineering.
Colossal's 2025 PGC breakthrough brings hope for reviving this iconic bird.
From culturing pigeon germ cells to chicken surrogates, de-extinction path is complex.
Will de-extinction restore Mauritius ecosystems with the dodo as seed disperser, or distract from conserving endangered species today?
Welcome to Dodo 2025, reviving an icon. Addressing the elephant in the room, our AI hosts deliver accurate science stories efficiently.
Powered by AI, we bring cutting-edge de-extinction updates, with all facts verified from reliable sources in biotechnology and conservation fields.
Colossal's breakthrough in pigeon PGC culture marks a pivotal step in avian de-extinction, potentially bringing back species like the dodo.
Yet, biological and ethical challenges persist in de-extinction. Our thesis explores how technology intersects with ethics in modern conservation practices. The dodo, Raphis cucullatus, was large flightless bird, endemic to Mauritius, extinct in 1681 by humans and invasive species like rats.
Colossal Biosciences announced in September 2025 a breakthrough in culturing pigeon PGCs, a key step toward resurrecting this extinct bird.
The dodo's extinction was due to hunting, habitat loss, and predators like rats, cats, and pigs introduced by sailors.
This breakthrough bypasses avian cloning limits, enabling edits for dodo traits in Nicobar pigeon and aiming for a self-sustaining Mauritius population.
Mauritius has changed. Careful reintroduction design must restore seed dispersal without harming today's ecosystems or competing recovery priorities.
Skeptics warn of simulated dodos and ethical costs. Rigorous monitoring and transparent standards are vital for biological integrity.
The project aims for a self-sustaining population in Mauritius, restoring seed dispersal. This is a significant test for biotechnology and conservation.
Biological challenges include interspecies surrogacy using sterile chickens. This is a complex process requiring careful genetic engineering and animal welfare considerations.
Timeline targets 5 to 7 years for a first hatchling. Patience, stable funding, and disciplined expectations are essential.
Scaling to thousands likely takes decades, underscoring long-term investment needs and sober expectations for de-extinction outcomes.
Once a symbol of loss, the dodo now tests biotech ethics and demands rigorous, transparent evaluation before any release.
Avian reproduction makes cloning impossible due to egg complexity. This is a fundamental biological limitation that de-extinction must overcome.
PGCs are key, precursors to sperm and eggs in birds. This is the starting point for genetic editing in birds.
Colossal screened hundreds of media and reliably cultured pigeon PGCs, a milestone proving feasibility for downstream gene editing.
Editing, then transfer into surrogate chicken embryos, advances a dodo-like phenotype through cautious, stepwise germline engineering in trials.
Chimeric chickens could produce pigeon gametes carrying edits. Validated integration precedes any limited, carefully monitored breeding phases in pilots.
Nicobar pigeon is the base, closest relative to dodo. This is the species used for the genetic engineering work.
Comparative genomics identifies genes for dodo traits like beak shape. This is the foundation for understanding the genetic basis of dodo traits.
Flightlessness and size are key traits to engineer. This is the primary goal of the genetic engineering work.
The project includes advisory committee for Mauritius rewilding. This is a crucial component for ensuring cultural sensitivity and scientific oversight.
Safe, rat-free release sites with community oversight are non-negotiable to protect birds and earn durable social license.
Dodo's role in seed dispersal could restore ecosystems. This is one of the key ecological benefits of the dodo's reintroduction. Diving into data, Colossal's PGC milestone overcomes avian cloning barriers, enabling germline editing for projects like Dodo De Extinction and more.
Birds can't be cloned like mammals because their eggs are complex structures. This is a fundamental biological limitation.
Egg development begins in the oviduct, making nuclear transfer unworkable in birds, a core barrier to cloning approaches.
By laying time, it's a blastoderm with thousands of cells. This is the initial stage of embryo development.
PGCs offer the alternative, as germline precursors in embryos. This is a promising avenue for genetic editing in birds.
They circulate briefly, allowing isolation and culture for editing. This is a critical step for the PGC culture process.
Long-term culture was limited to chickens until this breakthrough. This is a significant advancement in the field.
Colossal tested over 300 media to find the pigeon recipe. This is a meticulous process to optimize the PGC culture conditions.
Kells grew for 2 months, doubling every 35 hours. This is a key metric for the PGC culture process.
They migrated to gonads in surrogates, confirming functionality. This is a crucial step to ensure the PGCs are functional.
This enables CRISPR editing for dodo traits in Nicobar PGCs. This is the final step in the genetic editing process.
Sterile chickens are created by knocking out DDX4 gene. This is a specific genetic modification to prevent egg formation.
Edited PGCs are injected into chicken embryos to create chimeras for breeding and stepwise trait validation in vivo.
Chimeras have chicken bodies but produce pigeon gametes. This is the desired outcome, where the genetic material is successfully integrated.
Breeding chimeras yields the first dodo proxy offspring. This is the first step towards a viable dodo population.
Myth busted. De-extinction is impossible. It's now feasible with this tech. This is a key takeaway from the data presented.
Comparative genomics uses Nicobar, solitaire, and tooth-billed pigeon genomes. This is the foundation for identifying the genetic basis of dodo traits.
Identifying genes for beak, face, and flightlessness traits. This is the process of pinpointing the specific genes responsible for dodo characteristics.
CT scans of 75 bird skulls aid craniofacial editing. This is a crucial step for ensuring the dodo's appearance is authentic.
Interspecies surrogacy risks immunological rejection and developmental incompatibility, posing significant biological, welfare, and logistics challenges in practice today.
Egg size mismatch is a major biological bottleneck. This is a critical factor in the success of de-extinction.
Rewilding needs an independent advisory committee and safe release sites with phased monitoring and clear exit criteria from day one.
Dodo as keystone herbivore for seed dispersal in Mauritius. This is one of the ecological benefits of the dodo's reintroduction.
Ecosystem has changed. Consequences of reintroduction unknown. This highlights the complexity of de-extinction and its potential unintended consequences.
Myth: rewilding is easy. Reality: it's experimental, slow, and success depends on habitat, funding, and social license.
Data shows potential for happy ecological surprises. This is a positive outlook on the potential benefits of de-extinction.
Busting myth. Timeline is hype. First hatchling is proof of concept. This is a key point to emphasize the progress made.
Scaling population takes decades of breeding and monitoring. This is a long-term commitment required for de-extinction.
Venture capital drives ambitious but strategic timelines. This is a significant source of funding for de-extinction projects.
The platform could aid endangered birds via genetic rescue, biobanks, and assisted gene flow, complementing, not replacing, habitat work.
Debate time. Critics argue the revived dodo is merely a genetic simulation, lacking true authenticity of the original species from Mauritius Island.
Critics say authenticity fails without native behavior and full genotype, closer to a portrait of kin than true revival.
Moral hazard. The promise of reversal may blunt prevention if people assume technology can always fix losses later.
It can distract from habitat protection and climate action, driving present extinctions. The emergencies to fund first, right now.
Opportunity cost: dollars for one proxy might rescue many species. via habitat, rangers, biosecurity, and steady community work.
Where to put them? The original habitat is gone, so zoo curiosities at best. This is a practical consideration for reintroduction.
Animal welfare concerns, high embryo mortality, and surrogate suffering in the process. This is a critical ethical consideration.
Ecological hubris. Introducing engineered species could disrupt modern ecosystems unpredictably. This is a philosophical concern about the long-term implications.
De-extinction treats species as genetic traits, ignoring environment and evolution. This is a key philosophical debate about the de-extinction approach.
What do you think? Is de-extinction worth the risks? Comment below. For conservation purposes.
PGC tech could aid Philippine eagle and kakapo, species facing steep extinction risk without rapid, coordinated conservation action.
Funding for this research comes from venture capital seeking breakthrough returns. This is a significant source of funding for de-extinction projects.
Colossal raised $120 million to expand their de-extinction portfolio beyond dodos. This is a significant investment in the field.
Woolly mammoth and Tasmanian tiger projects use similar genetic rescue approaches. This is a comparison to other de-extinction projects.
The success metrics include first hatchling, population growth, and ecosystem integration. This is the primary goal of the project.
Critics worry about opportunistic narratives, overshadowing conservation urgency for living species. This is a concern about public perception.
Ethical oversight involves international wildlife committees and indigenous communities. This is a crucial component for ensuring ethical considerations.
Mauritius government collaboration ensures cultural sensitivity in dodo reintroduction planning. This is a specific example of a successful collaboration.
Understood. I'll deliver the script with a calm and informative tone, moderate pace, and clear articulation. Here we go. Public engagement campaigns educate communities about de-extinction goals and limitations. This is a key component for successful public outreach.
Scientific publications undergo peer review to validate PGC cultivation and editing claims. This is a crucial step for scientific credibility.
Global reproducibility across independent labs, not one company, will decide whether this breakthrough truly scales worldwide at all.
Timeline milestones include first chimera chicken, dodo proxy egg, and viable hatchling. This is a key milestone for the project.
Behavioral studies will test whether proxies forage and
Diversify into biotech startups focused on conservation and species recovery. This is a significant source of funding for conservation biotechnology.
This breakthrough represents humanity's evolving relationship with extinction and natural heritage. This is a reflection on the broader implications of de-extinction. Actionable takeaways. Evidence first. 1. Support habitat work that prevents extinctions now. Local land trusts, invasive control, and island biosecurity deliver the biggest near-term gains. 2. Add your eyes and ears to open datasets. Upload bird calls, trail cam shots, and reef notes with time and location so scientists can track change. 3. Pressure test big claims with sources. The September 2025 Pigeon PGC milestone matters because it finally enables editing in avian germlines. Without that, birds can't be cloned. But feasibility isn't destiny. Interspecies surrogacy may fail. Timelines get stretched, and ethics must be real, not marketing.
If we earned it, subscribe, like, and share this with the friend who always asks good questions. Then, tell us your favorite moment and one thing we should do better. We will pin helpful resources and your top comments. Remember, conservation moves when we pair courage with care. Plant something native, protect what is left, keep the receipts, and teach one person what you learned today, so we can celebrate together in the comments. If you are posting your first audio clip or native plant, tag us so we don't miss it. We will see you next time with a careful look at Arctic rewilding claims and what they mean for people on the ground. Until then, take a breath, touch a tree, and be kind to something small. Then, tell a friend why it matters, and invite them to join us next week for the results. We will pin a quick start guide in the comments for newcomers and a link to our sources/notes panel.